Hello, hello. Welcome to another episode of the Chemistry Senior Seminar Series. Adrianna is going to tell us about her research today, and Professor Stefan Debert is going to be doing the introduction.
Hi, friends. So I'm Stefan. It is my great honor and privilege and pleasure to introduce Adrianna Hudyma, who is going to be giving some pretty hardcore biochemistry talk with us. So Adrianna was in my crazy pandemic organic 2 class, organic 1 and 2, where we really asked a lot of our students in terms of being self-directed and just really chasing hard stuff. And I think that she got really good at that. And it was really impressive to see both her and her classmates respond to that challenge. And then for the last year, we've been just talking about this talk, right? So Adrianna came to see me in the fall and said, what should I talk about? And I said, I don't know. I don't know, man. And then we said, well, how about epigenetics? And I was like, I don't know what that is, but it sounds great. And we basically spent the fall term, I think even the winter term, really digging into what are the interesting questions here and what can we take away from it. And I think it's a great model that we ask our students to do, to really go and chase it down, right? And use this senior experience as an opportunity to get smart about something that you want to get smart in, right? So we're just kind of covering, and I really, really appreciate that. And so I'll let her display that hard-won expertise over the next half hour or whatever, and I'll cede the floor to her.
Thank you, Stefan. Okay. Does that help a little? Okay. All right. Thank you, Stefan. So as he said, my topic is exploring epigenetic approaches to cancer treatment. And I'm just going to start off by laying out what we're going to talk about today. We are going to start with some background on what is epigenetics, why should we care about this, and then we're going to move on to our first mechanism, which is DNA methylation, and then we'll talk about ways to inhibit DNA methylation, and then we'll switch gears and we'll talk about histone deacetylases, and then ways to inhibit our histone deacetylases, and finally we will wrap it up with where the field is now. So first off, why are we going to bother to study epigenetic drugs for cancer? We have chemotherapy, we have radiation, we have immunotherapies, we have tons of great things that work. So why bring in something else? And the main reason is cancer is adaptive. It's under really high evolutionary pressure to adapt to treatment conditions. And the longer it takes for a treatment to work, the more time you give for a risk of a random mutation that allows it to become treatment resistant. So we want to be able to target things quickly, and sometimes that requires a combination. And then also, sometimes cancer just will not respond to what we try to treat it with first, or it becomes resistant, and when we have resistant cancer, we need other options for treatment. So what actually is epigenetics? Epigenetics is reversible changes to gene expression that do not alter the genetic sequence itself. So you're still going to have the same sequence of A, C, T, G, whatever that is, but it's going to change how accessible that part of the DNA is. And that happens through mechanisms that include DNA methylation, histone deacetylation, and microRNAs. I will not cover microRNAs as there's no FDA-approved drugs that target that. And so we all are familiar with our double helix DNA structure, where you have base pairing between A and T. Those are the hydrogen bonds, and then G and C also hydrogen bond to create this double-stranded helix. And then between the different bases on the same strand, we have what we call a phosphodiester bond, which is just what is holding together the bases on the same strand. And so those are important things to remember. Unfortunately, that is not how simple it is in your cells on a daily basis. So this is what a little bit more of what the structure is going to look like when you actually were to look into a cell. So our little blue line is our DNA double helix, and this double helix is then wound around these histone proteins, which are those purple cylinders, and then a whole bunch of those histone cylinders are in a grouping, and that we call a nucleosome. And so if you were to be able to zoom in and actually look at the inside of the nucleus of your cell, what we would see is this grouping and this winding of that DNA around other things. It's not just hanging out in a little helix for us. And what I've been talking about is how accessible that is. As you can see, it's wound around all these things. And so there's two main things that we talk about when we talk about the accessibility of DNA. And the first one is, this is the two main structures. On the left, you can see active chromatin. Chromatin is just another word for your DNA. And this active euchromatin has plenty of room between things. There's space for all sorts of things to get in there and bind. And as a result of that, those genes are turned on and we get gene expression. And then on the right, we have this closed, condensed chromatin. Everything is really scrunched together. There's hardly any room for anything to get in there. And as a result, you have those genes turned off. They're not able to be transcribed. And then we don't get any gene products. So there's a very specific point that this is happening, those changes in DNA that I care about. And it's in this first decision-making window here. And that's the location of your oncogenes and your tumor suppressor genes. When a cell is big and healthy, it wants to divide and make more copies of itself. And so it reaches this decision-making window, and those oncogenes are just genes that say, like, yes, this looks great, we're big, we're healthy, let's continue and let's divide. And these tumor suppressor genes are the genes that are going to stop and say, are environmental conditions right? And then most importantly for what I'm talking about today, does everything in this cell look right? Do we have the right number of copies of everything? Is everything healthy looking? Have we made any significant mistakes? And if they detect any of those mistakes, they're going to pause this process. We will not get to replicate our DNA. Instead, they will stop things, and they, in most cases, will lead to cell death because this is not a healthy cell, and we don't want to make more of it. So when things happen to our tumor suppressor genes and they're no longer doing our job, this is often a cause of our cancer. And the way that that often happens is that the promoter of those genes is not visible. So I'm going to show gene structure. Like most things in science, it gets really complicated really fast. Most of this will not matter for our purposes today. The important thing is the promoter region of a gene is in front of the actual gene that gets transcribed, and that a bunch of these things all have to have the space to get in there to bind to the promoter region before gene transcription happens. So that's why we need that loose structure. We need space for all of these transcription factors to get in there and bind to that DNA. So again, cancer causes changes to important regulatory genes, and it makes them that heterochromatin, that really condensed chromatin, where you can't get transcription factors in to bind to the DNA to tell your body to transcribe that gene. And there's two main ways that your body is going to do that. We have DNA methylation, which is what I'm going to talk about next here. And then in the second half of my talk, I'm going to talk about histone deacetylation, which is our other mechanism. So here we have DNA methylation. This is your base cytosine, which is one of the 4 bases of your DNA. And typically, cytosine has this hydrogen at the 5 position right here. And then there's these enzymes called DNA methyltransferases, they're abbreviated DNMT, that come along and they catalyze a reaction, and it puts this methyl group onto your cytosine. And what this does is when you have a methyl group, that's a hydrophobic group, and so you induce these hydrophobic interactions, and hydrophobic groups in a protein prefer to be folded into the inside away from the liquid in your cells because that is a polar liquid, and they are nonpolar. So this happens in a very specific place inside of your cells. We call these CPG islands, and that means that there's a cytosine base, the phosphodiester bond I mentioned on that first slide, and then guanine, which is another base of your DNA. We call those CPG dinucleotides. It's important to note that it has to happen on the same strand. So these are the correct way. We have C, P, and G. happening on both strands. And it must be on the same strand, so there's that phosphodiester bond between the two of them. Whereas this is not right. This is not going to cause that reaction because the C and the G are on the opposite strands of DNA. And then if there is a whole bunch of these CpG dinucleotides, this is going to be called a CpG island in most of the literature. And it just refers to an area of DNA where we have lots of these CpG dinucleotides. And these are hemimethylated, so if we have a methyl group get put on this C here, we're going to get another one that gets put on this C down here. And then it's important to know that these are pretty rare in our genome. So this is cytosine, and this is thymine, which is another base of your DNA. And so methylated cytosines look very similar to thymine, and over time, through many generations of cell division, your body will make mistakes. We can see if we were to draw in a methyl group on our cytosine that it now looks very similar to our thymine. The only difference is this amine group up there. And so that looks really similar to your body, and it's more likely to make mistakes that it does not catch, leading to them being statistically rare in our genome. And those remaining ones happen at more specific places. Specifically, we would expect to find them in the linker DNA, which is between that big complex that I showed you earlier. As again, we want that DNA to be loose and able to be transcribed, and there's plenty of space when it's not wound around those other proteins. So what are the structures of the drugs that actually inhibit this mechanism? These are the two FDA-approved drugs. They're approved in 2004 and 2006. And I'm going to pull up the base cytosine, which is what we're trying to inhibit, getting methylated. And as you can see, they look very similar. The only difference is this nitrogen here at the 5 position of the ring, which changes how your body is able to catalyze the reaction to put the methyl group on. And then the key difference between azacitidine and decitabine is this extra OH on the azacitidine. This makes it a ribose sugar base, which is what is in your RNA. And as a result, you get this drug incorporated into both your RNA and DNA. Decitabine does not have that OH, and so it can only be incorporated into your DNA, which is deoxyribosome, so one less oxygen group there. A little bit more about these. They were discovered in the 1960s as classical cytostatic agents, and that just means that it was found that they inhibited growth of cancer, but they were not cytotoxic. So they were not able to kill those cancer cells that they initially looked at them for. And then it was later realized that these compounds were able to inhibit DNA methylation in humans, which was an exciting development. And that is why they were FDA approved to treat specific cancers in 2004 and 2006. So how does this actually work? What is this drug doing once it is into your body? It's being incorporated directly into your DNA instead of your native cytosine base. And once you have this into your body, there's a step that has to happen first. They're called prodrugs. And so you need this ATP-dependent phosphorylation to get a monophosphorylated form in order for your body to be able to recognize it and put it into your DNA. And so we are going to go over the actual mechanism of this drug next. On your left, you will see cytosine, which is your normal base in your DNA. And on the right, one of these drug analogs. Again, this key difference is that nitrogen there. So we're going to follow along with that. It won't matter in the first steps, but pay attention to it towards the end of this. So in this first step, we get this atom on the sulfur attacking the 6th position carbon of the ring. And this happens the same in both your body and in the drug. And then there's movement of electrons through those aromatic bonds. And in the second step, we have an acid-base reaction with that negatively charged oxygen. And again, we're going to move through those aromatic bonds. And then in this third step, this is where that nitrogen starts to matter. There's supposed to be an E2 reaction with the hydrogen on the 5 carbon of the cytosine. And it's an E2 reaction with something on the enzyme that is not drawn in this diagram. But in the case of the drug, we had a nitrogen there instead. It doesn't have a hydrogen as it already had 3 bonds without the hydrogen. And so this last reaction cannot proceed. And this last reaction is what releases your DNA from the enzyme. And now it has that methyl group on it. Whereas with this drug, it can't release the enzyme because it can't do that final reaction. And your body will recognize this as incorrect, and it gets very concerned, and it will just choose to degrade the entire complex, including your enzyme. And so at low levels of the drug, what happens is you just get a decrease in the amount of enzyme in your cells. And so they're not able to methylate the genome at the same level. And in the next generations of cells, you restore normal cell markers, your genes will get turned back on, and you will have dealt with some of those cancer cells. And at high levels, it happens to stall the replication fork, and what happens is that cell will actually die. So that would be how it works. And then we want to prove that it's actually doing this in our body. So they did a lot of proof of concept experiments. This is a pretty common type of way to test things in biology. They took cells with an antibiotic resistance gene, and they made the promoter region of that gene methylated, and so it's that 2-type form. You can't get anything in to bind, so they can't use that gene anymore. And then they poured plates that have an antibiotic in the plate medium, and so the only thing that can grow is something that has resistance to that antibiotic. And as you can see, when they plated those cells with no drug and the antibiotic present, nothing was able to grow because that gene has the promoter region methylated. And then they plated those same cells on a plate that did not have the antibiotic, and everything was able to grow. And then they plated the cells with the gene on plates with both the antibiotic and our drugs. And so if our drug is working, we would expect that colonies will grow because the promoter will no longer be methylated. And as you can see, we get more and more colonies as we're increasing the concentration of drug. So we can tell that it is doing what we expect. And zebularin is just another one of these DNA methyltransferase inhibitors. And here is a second way to look at that. This is a graphical representation of it, and what they did is they looked at the percentage of the promoter regions of this tumor suppressor gene that were methylated once we introduced the treatment. And you can tell that as we increase the amount of treatment, less and less of those promoters are methylated, which again is showing that our drug is doing what we expect in the body. And then there's a third way to look at this. Again, P16 is our tumor suppressor gene. And so if that's methylated and the promoter is, then we don't expect our gene product. And these blots are the gene product. And so as we increase the concentration of our drug, we're getting more and more product from this gene, which means that the promoter is no longer methylated. And then that bottom row with the GADPH is just a loading control. This was a gene that was not affected. And so we would expect the same amount in each one. And that's just to prove that we put the same amount of sample in each position and didn't just add more to get these results. So now that we have proven that this is actually doing what we expect it to do in cells, we wanted to see if it targets cancer cells over our normal cells. And so this group did an experiment where they took cancerous lines of T cells. That's the graph on the left. That's just 3 lines of cells with cancerous properties. And they put them in various concentrations of decitabine, which is one of our drugs. And then they measured the percent of cells that undergo programmed cell death, which is that percent apoptosis on the y-axis. And as you can see, there's a pretty high level of cell death when we get up to about 20 micromolar concentration of our drug. And then on the right graph, we have our healthy T cells, which is what we do not want to target. And there is some cell death, but it's a much lower level than we see in the cancerous lines, which is good. We want to make sure our drugs are targeting our cancerous cells as much as possible and leaving our healthy cells alone. So far, everything looks great. We have drugs that inhibit this. We know how they work. We've proven that they're actually doing what we want and that they target our cancerous cells, but there are some limitations. Most notably, they have an incredibly short half-life, and this is for the most part due to an enzyme in your liver and spleen that breaks apart cytosine analogs, and so once you break it apart, it can't be incorporated into your DNA, and so then it was not able to do its job. These worked best on hematological malignancies, so these are cancers of your blood. They didn't get many results with solid tumors. And then it's been very difficult to create oral formulas for these drugs, which makes them a little bit less useful in the clinic. And most of that came down to the really short half-life. Again, these were approved in 2004 and 2006. It took until 2020 for oral formulas to come out for either of these drugs. So it took a lot of more time engineering them to make that a possibility. What actually are these approved to treat? There's two main cancers. Our first one is this myeloid dysplastic syndrome, or MDS, which is a group of cancers in which immature blood stem cells do not properly develop and differentiate into different blood cell types. Instead, they either die still in your bone marrow or shortly after they are released. As a result, they're dividing quickly. They're taking up the space in the marrow, and so your healthy stem cells are not able to grow and divide and give you the correct products of cells in your blood. And then patients are experiencing anemia, shortness of breath, risk of infection, and this can progress to adult myeloid leukemia. And this happens primarily in older populations, especially those that have already had to undergo chemotherapy or radiation in their lifetime. I'm going to pull up a clinical trial that was with azacitidine. That top blue line is our azacitidine, and the bottom line is our conventional care. On the x-axis, we have the amount of people that are surviving out of a percent or proportion, and then on the x-axis is our months of treatment. And as you can see, at any given point, our azacitidine is keeping more of our patients alive, and there's a really low p-value, so we know that these are statistically significant results. So this is really exciting. This drug is actually working better than what we would usually choose to treat cancer first. And then they also have found that if you have patients that are strong enough to undergo chemotherapy, because this primarily targets older populations, a lot of people aren't able to tolerate chemotherapy, and so this can be used instead because it's a little bit gentler. But if they are able to tolerate that, what we find is if you can induce a complete remission with chemotherapy and then switch them to azacitidine, they are able to maintain their remission for much longer. And so it's a little bit gentler way if they're not able to go all the way through a course of chemotherapy, but we still get better results for those patients. And then the second disease that these are able to treat is myeloid stem cell, adult acute myeloid leukemia. Your myeloid stem cells are, again, not developing correctly, and they're taking up the space in your bone marrow, and then they cannot develop normally. And so patients are experiencing fevers, infection, anemia, and easy bleeding. And this is a really fast progressing cancer, and so we want to make sure that we get people treated quickly. So where are we now? We have covered our background, and then we covered DNA methylation. How does that work? And then we talked about ways to inhibit it and the cancers that can be treated by doing that. And now we are going to switch gears completely and talk about histone deacetylases and histone deacetylase inhibitors. And then where's the field now? So we're going to go all the way back and talk about another mechanism, and that is how chromatin structure is changed by histone deacetylases, or HDACs. If we look here up at the top, we have our big, loose, open DNA structure, and there's these acetyl groups on there. That is because their histone tails have the residue lysine in them, and lysine is a positively charged amino acid under physiological conditions. And because it has this positive charge, the negative backbone of DNA is going to interact very tightly, or bind really tightly to that, and we get this chromatin compression. Everything's tight. It's in that heterochromatin where we can't get transcription factors in to bind, which isn't ideal for trying to use those genes. So if we put an acetyl group on, and that's done by an enzyme called histone acetylase, then that negative charge or positive charge of the lysine has this protecting group on it. It's no longer able to interact with the DNA as strongly, and we get nice loose DNA. But HDAC will come along and it will pull that acetyl group back off and compress your DNA structure by inducing that strong interaction between the histone tail and your DNA. I know that's a lot, and so this is just looking at that again. On the top, we have those positive lysine residues of the histone, and they're hugging that DNA really tightly because there's a positive and negative charge. And then when we add an acetyl group, we have hidden that positive charge on the lysine, and so it's neutral and a negative charge. They don't really care about each other, and you can see that that has loosened that blue DNA around our histone protein, and there's space between the DNA and the histone in which we can get transcription factors in there to bind. So there are 4 drugs that have been FDA approved to treat cancer by inhibiting that mechanism. And we are going to start with SAHA, which was a big deal in the early 2000s. We'll again look at kind of a proof of concept. So in this situation, they took 2 cancerous cell lines, and then they plated them on a plate with either a control, which they did not expect to affect the cancer cells, or our drug of interest, SAHA, which they expected would inhibit growth and kill colonies. And every little purple dot is a colony on those plates, and so you can see across that top row with our control, there is lots of cancer cells. And then on the bottom with our drug of interest, there is very few cells, and they're a lot smaller. And then that little tiny graph on there is also a way to look at this in a graphical representation, and so you can see there's a drastic drop-off in the number of colonies of cancer cells that were able to grow in our drug. A little bit more about SAHA. It was approved in October of 2006. It's used to treat a rare cancer called cutaneous T-cell lymphoma. It's been approved only to treat refractory cancer that has already failed 2 systematic therapies. So like I was talking about at the beginning, we have cancers that will fail chemotherapy, they'll become resistant, and these drugs can then come in and help. And it inhibits by inhibiting zinc-dependent HDACs, which is most of the HDACs in humans. And that just means that there is a zinc atom that binds to the enzyme in order for it to be able to do its job. Moving on to our next one, we have poanobinostat, which was FDA approved in February of 2005. It's approved as a combination therapy for recurrent multiple myeloma, so it's going to be used in combination with another medication. And it's currently being investigated for quite a few other number of cancers and even some genetic conditions. We're going to take a look at some of the clinical trial data from this. So this was the study that got it approved. I know this is a really busy graph, and so on our y-axis we have progression-free survival probability. So basically how long are we getting patients that are able to live with this drug combination. And then our x-axis is just the number of months. And that top line we see is our combination therapy, and the bottom line is the same 2 other drugs and then a placebo. And so I'm going to draw your attention to this, because this is what is most exciting about this. The combination that includes poanobinostat gave a median progression-free survival timeline of 12.48 months, so over a year, which is really significant. And if we give the same combination of drugs, but instead we use a placebo, we only got 4.7 months of progression-free survival. So the addition of our drug improves how long patients are without progression of their disease for another 8 months, which is really significant. And then again, we have this really low p-value, so we can feel confident that these are statistically significant results. Moving to our third medication, we have belanostat, which was approved in July of 2014 for both relapsed or refractory peripheral T-cell lymphoma. And it again is going to broadly inhibit all zinc-dependent HDACs. And it was well tolerated in clinical trials, which is exciting. I know I've said that these are a little gentler than chemotherapy, but they are still cancer drugs, which tend to have really significant side effects for our patients. And so finding one that was a little bit more tolerable is good news. and then we're going to look at some clinical trial data from this as well. The median overall survival was 7.8 months. That's our y-axis. And then again, we have time on our x-axis. The thing that is most exciting about this, though, is if we look here, from 22 months to the end of the trial, which was 10 months later, 25% of patients are surviving, and that's holding steady. So for that 25% of patients with this disease, treatment with this medication induces a really long amount of time in which they're responding to it, which is significant. It would be something we want to try then, because it might be all it takes for those patients to have many months of time. This is almost a 3-year trial, so that's almost 3 more years of life in a disease that was already refractory, and so those patients were in a bad spot. And this is our last medication, robitussin, which was FDA approved to treat the cutaneous T-cell lymphoma in 2009, and then is also approved to treat the other one, which is our relapsed or refractory peripheral T-cell lymphoma, and that was in 2011. And what's really interesting about this one is it is under investigation for some solid tumors, which is something that a lot of these were not able to do. And it's showing promise as a combination therapy. I know I mentioned at the beginning, sometimes we want to take multiple medications and give them at once, as cancer is adaptive and we don't want to give it time. So because this is really interesting, I am going to show you one of the early ones for solid tumor. This is just in a mouse model, and it was looking at colon cancer. And what they did is they took mice that have colon cancer growths, and they injected them with either placebo or robitussin, and then they waited and looked at how big those tumors were 30 days after the medication. And on this first side that says A, we're talking about tumor size and volume. And you can see that red line is significantly smaller in volume, so it was working. And then on the right, that bar graph is looking at our tumor size in grams. And you can see there's a significant reduction in mass, a little less than half, which is huge. Recently, there have been some stage 1 clinical trials for solid tumors using this medication, which is, again, exciting and not something the other ones have been able to do. And then I mentioned that it has been used in combination therapies. So I am pulling one combination therapy trial. This one is especially interesting because it's taking azacitidine, which was our DNA methylation inhibitor that I talked about at the beginning, and then this one, which is an HDAC inhibitor. So this is using only epigenetic drugs, and you can see for those with T-cell lymphoma, there was a really drastic response. We see over half of our patients having this response to progression-free survival for quite a number of months. We're getting close to 2 years on that graph A. And then we're also seeing the duration of response is really long for those with T-cell lymphoma, which is really exciting. And it's really interesting that they are able to induce this level of response by only targeting with epigenetic drugs. There's a lot of other trials, though, that are taking combination therapies. And that would be more of our traditional chemotherapy or radiation in combination with these. So I know I threw a lot at you, so let's wrap it up with our key takeaways from this. Epigenetic drugs do have a role in cancer treatment. They have worked best on hematological malignancies. There's not as much data to support their use in solid tumors. Their success has been somewhat limited, but they've had a large impact for rare cancers. For example, that first one I showed you, MDS, where treatment with our azacitidine was actually better than conventional chemotherapy, which is exciting. And then they're also having really strong effects for rare cancers that had refractory disease. So where we were out of options for these patients, and now we have other options, which is really important for them. And then they have shown promise at augmenting existing therapies. And this is probably where most of this field is heading. There is papers you can pull where they are talking about 30 different ongoing clinical trials that are combinations of one or more of these drugs with chemotherapy, radiation, or immunotherapy. And so they're getting really good results there. They're finding that adding one of these alongside of our traditional methods is giving better results to patients without any negative effects. So that's where this field is heading. That's really important as, again, cancer is adaptive. All right, and I'd like to thank Stefan Debert for doing this project with me, and then Professor Kelly Colhane for answering all of my biochemistry questions as I did this, and then Professor Beth DeStasio for her genetics and molecular biology expertise, and then Professor Rachel Daughtry as our Chem 680 professor, and then my classmates for all of their feedback.
All right, our speaker would be happy to take questions if anybody has any for her.
I did. What how are most people getting the drugs?
What?
How do you get these epigenetic drugs?
Typically, you would have to be in a position where your doctor is like, we're stuck with options, and so then they would direct you to clinical trials, as there is no longer a standard care plan for you to follow, and instead your best option is to try something that's under investigation.
But how do you actually get them to the
You would get enrolled in a clinical trial in, like, usually one of the big research centers, and then they would be in charge of coordinating your care and give you a prescription for these as a part of the clinical trial.
Orally or through your blood, or?
Depends on the medication. Those first two I talked about, they were intravenous drugs for up until 2020. The goal usually is to get an oral route just because that's a lot easier for patients. It's not always possible just due to the drug structure and stuff.
Other questions? Sam?
Could you go back a few slides to the graph with the T-cell lymphoma versus non-T-cell?
The combination?
Yeah. Yeah, so I was just curious, what is like the non-T-cell lymphoma line representing? Like, is it representing those drugs on cancers that aren't T-cell lymphomas? I just wasn't sure what that line represents.
My understanding is lymphoma is just a broad category of cancers of the blood, and so T cells is a specific type of cell in your blood, and that would be if it was targeting a different type of cell in your blood.
Okay.
One last question. What causes the side effect? What, like most people you hear about with cancer treatments, some have really bad side effects. So, so the way I understand it, these epigenetic drugs are introduced, and then they're supposed to trick your cells into getting rid of the bad cells and making new cells. So where in that process is it so hard on your body?
For the first ones, those DNA methyltransferase ones, I talked about the enzyme in your liver and spleen that like breaks them down, and so your certain organs in your body do most of that breaking down of random stuff, and so they are usually under a lot of stress with whatever drug you've introduced to the system, and then most drugs, as much as we try and make them specific, they sometimes have a little bit of an off-target effect that we didn't expect.
Okay.
Hi, let me know if you can't hear me. In one of your studies, you said 25% of the patients were still doing quite well several months after, and I was wondering if you came across any reasons as why certain patients might be more susceptible to this treatment or might get better effects from it.
I don't know exactly why these 25% in this specific trial. However, for all of the cancers I talked about today, they have identified mutations in the genes that encode the enzymes that we're targeting as a higher prevalence than the normal population. And so they think that's why these drugs work so good on these cancers and less good on our other cancers and our solid tumors.
I'm not sure if you're going to hear me either, but when you said that these drugs were gentle, I just wanted to get a clarification on that. Is the gentle that they're still specific for the cancer, but it's not as painful for patients compared to chemotherapy, or they're not, they don't attack the cancer as aggressively as chemotherapy would?
The one where I said that, when they had patients in that clinical trial, those patients were reporting less side effects and less significant side effects. So they gave side effects a grade, and so they're having less grade 3 and grade 4, which is those really concerning side effects. And so that gets called less gentle. It definitely does not mean that it's a fun experience to take that medication, though.
with the DNA methyl inhibitors, I was curious if you knew what changes needed to be made to the structure of the drug in order for them to be made into oral medications.
That I honestly do not know. I didn't look into that as much. It was something I was just excited to see, but I'm not sure. I'm sorry.
Other questions for our speaker? If not, let's thank her again. On-campus folks, join us next week for even more of the fun. 2 more next week and 4 more the week after. Thanks for coming.
Yeah, no.